Hello class. We are going to cover the prokaryotic cell structure. And so right on the top of this part of the worksheet, just erase this. And this is going to be prokaryotic, which includes the bacteria and archaea. Okay, so again, prokaryotic cells do not have a nucleus, and so they do have specific things about them to protect their cell, such as a cell wall, different types of cell membranes. So we're going to talk about the cell wall for gram-positive, gram-negative bacteria and archaea. So the cell wall, we're going to put the function over here. So the cell wall functions to protect, right? So it's always next to, so it's always outside, right outside the cell membrane or covering the cell membrane. So it covers the cell membrane. And then from when it covers the cell membrane, it's going to essentially protect and keep the shape of the cell. Okay. All right, so that's the main function for all cell walls and all cells. And how gram-positive versus gram-negative have their cell wall is, as we talked about in class, gram-positive have a thicker cell wall, and it's made of peptidoglycan. We're just going to put a barrier there. So it's made, well, let's do, made of, and I'm going to do this in two colors, peptido, because that's the peptide part, and then the glycan part we're going to put in pink, because that's the sugar part of the cell wall. So there's two parts to the cell wall for gram-positive, and it's just more layers of the peptidoglycan. So for gram-positive, they can go up to like 25 layers, right? So, or sorry, 15 layers. So gram-positive have around 15 layers of this peptidoglycan. And those layers of peptidoglycan essentially make the cell wall thicker. So I'm going to talk about the structure of the peptidoglycan, and then I'll go to your slides and you can look at what it actually looks like around the cell membrane. So the main structure that repeats is the glycan part, and then it's called NAM and NAG. So this is like N-glucosamine. You don't have to know like the full name, but just know that these are sugars, right, or carbohydrates. It's a disaccharide. And that means that it's the same two sugars that repeat and kind of make a chain with each other. So it'll always be NAM and then NAG. And then, yeah, so that's one layer of the sugars. Let me make sure I'm pouring. Okay. So then we'll draw another layer. And let me move this a little bit. Make it a little smaller. Okay. So we'll have another layer of the NAM and NAG. So I'm going to draw one more layer. Kind of below that, another chain. This is not really like, this is the layer of sugars, and another NAG. Okay, and then what connects these chains of sugars is the peptides. So there's going to be like little amino acids, and we draw amino acids like circles. Okay, so this shown right here, oops, this right here is called our amino acids, right? So those NCC, and they're cross links, right? So they are cross links. So this is going to, these right here are going to link the sugar, the lines of the sugars, right? The NAM-NAG chains, disaccharide chain is going to be linked with these cross links. Okay. And then that is basically what makes up the cell wall. So it's almost like it's a lattice, right? Like you would think of like a gate. So these little chains, if I like zoom out, it looks like this. 2 chains, and then the links that connect them. So if I look at this, it's that, right? And then this is like a lattice. And so these can be stacked on each other. You can have multiple chains of sugars and then cross-linking. And so you can see how basically protective this is, to be surrounded by this chains of sugars and the cross links. It's like a real fence that's surrounding the bacteria and protects it as a cell wall. So same kind of thing, exact thing for gram-negative, but we're going to add gram-negative extra membrane, which would be the outer membrane. So gram-negative, right outside the cell membrane, it's going to have a thinner cell wall. And it's going to still be made of peptidoglycan. Oh, let me write the colors. And you're still going to have the same exact thing, NAM sugar connected to a NAG, and then another NAM, and it can keep going for a long chain all the way around the cell. Okay, and then that will be cross-linked the same way. It's going to have less of these chains, which I'll show you in the slides. Okay, so I'm just drawing those out in NAG. Okay, and then we're going to connect them with this cross-link. Okay, so this whole thing will be thinner in the sense of like there'll be a less of the layers of these NAM-NAG chains and the cross-links. So this will lay on top of another NAM-NAG chain layer. So I'll show you kind of the perspective that I'm talking about. So if we look at the cell wall here, and we're going to look at gram-positive versus gram-negative. So we call this an envelope, right? All the layers that surround, the main layers that surround bacteria, gram-positive or gram-negative, we call it a cell envelope. So this whole structure here and this whole, all 3 of these layers here, this is called the cell envelope. Okay, so let me zoom in. So as you can see here, we're going to have gram-positive right here and gram-negative over here. And there's 3 layers in the gram-negative and just the 2 layers in the gram-positive. So what we're looking at here on the gram-positive is this whole layer right here is peptidoglycan. Okay, and so you're going to have those chains of sugars in here, and like this kind of, and then, oops, let me do the other color. So we're gonna have chains of sugars, and then you're gonna have cross links kind of going all in. So you can see this is like a lot of layers, and they're all kind of clumped up together, and this is all peptidoglycan. Right where the cytoplasmic membrane, right outside the cytoplasmic, covering the cytoplasmic membrane. And then on this side, we have the gram-negatives, and this one, you can see here's the membrane, same like gram-positive, but then it has a bit much thinner peptidoglycan. So basically, this one will be like 2 to 3 layers thick of peptidoglycan cell wall. Okay, so if we're gonna zoom into this one in another, but then you'll see that there's this extra outer membrane because the thin cell wall is so thin right here, right? This is all the cell wall. Then the gram-negative do have this whole outer membrane, and we'll talk about what these are that are sticking out of this membrane right here. Okay, so as you can see here, we have a gram-positive, right, in purple, stained, and then right here you have gram-negative rods stained pink because of the thinner cell wall, and then the thicker cell wall of gram-positive is going to stain purple. And then this is an actual electron micrograph picture of the peptidoglycan right here, and then cell membrane. You can tell this is gram-positive because it's only 2 layers, right? And then on this side, we can tell that this is gram-negative. So this whole thing's gram-negative because we have this thin little cute peptidoglycan right here, and then you have the cytoplasmic membrane, and then you have the outer membrane. So there's 3 layers. So that tells us it's gram-negative. Okay, so let me show you the gram-negative and the actual like just cell wall structure. So this is what I drew, right? This is G and M are NAG, G, and then M is NAM, okay? And they're repeated and they're bonded together. Oh, let me do in the pink because we did it in pink on the worksheet. So we had NAG, and then this one's NAM, okay? And then these are the cross links. So these are amino acids. And they're, oops, wrong color. So these are the amino acids right here. Okay. And then this is they would be the cross links, and you can see how it's linking this sugars to this sugar line. And then what we're looking at now is it kind of on a flat, right? Looking from not like the side of it, but like the top on top. And so then you can see the peptide cross links in blue, and then the lines of the sugars, the NAM and NAG, are in purple. So if I stack a bunch of these, right, then that's making one each layer of the peptidoglycan. So I'll show you that here. And so you can see here, this is a gram-positive bacteria, and you see all the layers of peptidoglycan. See how many layers. So it's like really thick amount of layers, right? So this is so these purple are the NAM and NAG, and then the blue is the cross-linking. And so that offers the bacteria so much protection. Being a single-celled organism, it really needs this protection. Okay. And then one thing I wanted to talk about is the types of interbridges or the types of cross-links can be made, can allow the bacteria to be more resistant. So this was showing Staph aureus versus E. coli, the cross-links here. And what you can see is like the type of the amino acids, right, of that species that cross-link the sugars, will change its ability to be killed easy or not. And what's interesting about Staph aureus, this is the flesh-eating bacteria, is that this one right here, see how much, this cross-link right here, see how many glycines and like connection the interbridge has? A lot more amino acids than E. coli. So you have like only 8 amino acids, and then this one's like has these huge interbridge of 5 glycines, which then create a stronger bridge, which makes it more resistant to things like penicillin. So that's why we often get MRSA, right, which is methicillin-resistant Staph aureus, because of this interbridge is very strong, right? And there's a lot of amino acids, and so that makes it more resistant to penicillins, or all the cillins, really. And if you're allergic to penicillin, then this one is methicillin. Methicillin. And so it's very easy for the bacteria, if they're resistant to one penicillin, then it will more likely get resistance to the other psyllin antibiotics. Okay, so this is why not all species, right? Different species are going to have, yes, they all have peptidoglycan, but they have variations of these amino acids, and that's going to cause them to be stronger and often more they hurt us more because of that when they're harder to kill. Okay, so then there are other things in the gram-positive, and these long things sticking out. These are called lipotechoic acids, and those are going to basically help. It doesn't say that, but so those are going to help like cell communication for gram-positive. They're like little antennas. That's not all they do, but just that's one thing they do. And they're part, like inside, embedded into the cell wall. Okay, and then remember this, reminder, remember, lysozyme. It's supposed to destroy the cell wall, and it's found in our human secretions, mucus, saliva, breast milk, right? So this little protein is found in our human secretions, our enzyme, and it will cleave the linkage between the sugars. So lysozyme will go in between the NAM and the NAG, right? The sugars, and then lysozyme will cut it. And so then that makes our spit and all these secretions with this enzyme be a little antimicrobial, so it can kill some gram-positive bacteria. All right. So then the last one is the Archaea cell wall. So this is a wall called pseudomurin. Okay, and these Archaea cannot be destroyed by lysozyme and penicillin because they have a different kind of sugar, and I'll write that now in your workbook. So Archaea have a cell wall of pseudomurin. And that cell wall is basically NAM, different sugars. So NAM and NAT. So a little bit different. Let me put a different color. So different sugars that are linked, but kind of same kind of concept. So lines of these sugars kind of repeating And then it will have like different types of cross-linking. And this is for Archaea. And then these Archaea can survive in the gut if it's like a methanogen bacteria. So there are ones that live in us. Oh yes, and then I also forgot to mention that gram-negative bacteria is only 2 to 3 layers of the peptidoglycan. And if we measure those layers, it's going to be around 8 nanometers in thickness. And then these like 15 layers is going to be around 20 to 35 nanometers, like if you actually measure it.